So can I have the next slide, please? I can't even operate, right? My slides. If you can bring that here, I can operate, right? Why won't come? Yeah, so what can I get the next slide? So in organizing the various kinds of activities in bioinformatics, the Department of Biotechnology has played a leadership role. And this has started from about the late 1990s, but really in a big way from about the 2000. Next slide. Traditionally in India, structural biology has had various kinds of strengths across multiple universities. And I'm not going to tell you the details or the history why structural biology has had strengths, but the structural biology actually has spawned various kinds of bioinformatics algorithms and bioinformatics research in various parts of India. And the research, again, I mean, this is not, I've just picked out a few, the areas that, the kind of approaches that have been taken in devising these algorithms and trying to solve various kinds of quantitative problems arising out of initially structural biology and then into other areas, including genomics, has been mathematical, then machine learning procedures more recently, and can I have the next slide? Statistical procedures and a hybrid of statistical and computational methods. So those have been the major territories or major approaches that have been taken, not just in India, I suppose globally as well, but we in India have also talked about these various kinds of approaches to bioinformatics development and solving various quantitative biological problems using bioinformatic and various kinds of approaches. I'll have the next slide. Many of these approaches have actually been driven by certain flagship projects that were, again, announced and supported by the Department of Biotechnology. And two major flagship projects come to mind. One is on tuberculosis, again, looking at the diversity of mycobacterium and in various geographical and clinical contexts, and the other is sequencing of the rice genome. And these two consortium projects actually led to the development of various kinds of algorithms, as also spawned a large number of centers of excellence and distributed information centers. These centers of excellence grew around the work that was being done in biology. And as you can see, New Delhi, Pune, Madurai, Bangalore, Calcutta, etc. So they were geographically spread as well. As these centers of excellence that were amassing the data that were generated in their regional areas on TB, rice genomics, and so on and so forth, the information that was being gathered had also to be dispersed across various institutions, researchers in other institutions. And so the Department of Biotechnology set up these distributed information centers that were established with the task of providing discipline-oriented information to all institutes. And these were not focused or constrained by the flagship consortium projects, but also other kinds of projects that were taken up. So we had centers of excellence come up in bioinformatics. We had distributed information centers come up in the area of bioinformatics. And then subsequently, there were various teaching programs that were initiated. And these included master's programs, PhD programs, short-term training programs, more in the way of summer and winter schools and workshops. And then the Department of Biotechnology also started a national certification examination, essentially to develop skills among a large number of people who would code the various kinds of requirements for the major centers of excellence or other departments in universities, etc. There was also around that time, from about the mid-2000s to about even till now, various kinds of molecular modeling are being done, and these molecular modeling needed visualization tools, etc. So there were some national facilities on interactive graphics and molecular medicine modeling that were set up, and these again were distributed throughout the country, as you can see: Bangalore, Hyderabad, Madurai, Pune, Calcutta, Delhi, etc. So they were geographically distributed. These were national facilities on visualization of quantitative data, especially with respect to molecular models. Again, these were primarily driven by structural biology, but also other kinds of biological information, biological data were being modeled. The strengths in bioinformatics, again, this is a personal view. People can add on during the panel discussion, etc. I think the strengths in bioinformatics stem from the fact that India has had a very, very long legacy in high-end mathematics or deep mathematics and statistics education, which are some of the core foundational areas in bioinformatics. Also, subsequently, a lot of good things have happened in computer science, including algorithm development, programming, and more recently, various kinds of degree programs that came up in various universities and also in institutions. So those have been the core strengths that India has in programming, algorithm development, mathematics, and statistics. However, having said that we have strengths, we have also faced lots of challenges, and one of the major challenges that I think that we face even till this day is that our exposure to hands-on new technologies come in late. We are at least 10 years behind schedule, behind the introduction of new technologies, and therefore, by the time we get introduced to these new technologies, many of the algorithms have already been developed. One reason why we have late hands-on exposure, of course, the time is shrinking, but still, even today, with respect to next-generation sequencing, our exposure to new technologies was delayed by at least 5 to 7 years. The new technologies are usually initially very expensive. Of course, the costs drop, and that's one reason why there is this phase lag in our exposure to these new technologies, and that creates a lot of problems because as the new technologies are being used, there's a demand on algorithms, but by the time we get to know that these algorithms are in demand, many of them actually have already been resolved or the methods are already available. The other challenge that I think is that we have a reluctance to move out of tradition in terms of course design, etc. in the universities. These new bioinformatics methodologies require also changes in course design. It's become very much multidisciplinary, but generally in our university curriculum, we are traditional, and therefore it's not easy to get our courses to change. It's easier in the national laboratories, but national laboratories cannot most of us cannot actually award degrees. So designing new courses becomes difficult, and that I believe is a constraint or a challenge that one needs to overcome. And also there is a reluctance to move out of our own traditional research. So it's not easy for many of us to move out of our comfort zones and sort of cut across disciplines and boundaries, which is a requirement in bioinformatics to be able to understand what the problems are and to use our strengths in order to develop new algorithms. So it's my belief that this particular meeting, which is again, with respect to India's gaining associate membership of the EMBL, engagement with EMBL may help overcome many of these challenges because the phase lag that we have with respect to our exposure to new technologies might actually be cut down because of exposure or our access to these core facilities that are usually set up very quickly as the new technologies are introduced, or many of the EMBL labs actually work with the industry in order to introduce the new technologies, and I think that's a major gain that India will have. Also, in order to design new courses to be able to think in a multidisciplinary way and work with the international community. I think those are very, very helpful. Again, during the panel discussion, more ideas may come, but I think that our engagement with EMBL will actually help us in a big way in this area of bioinformatics because of the exposure. We already have some engagement with bioinformatics. The National Institute of Biomedical Genomics already has done workshops with the EBI in the area of bioinformatics, and that, I must say, was an extremely helpful thing to begin with, because as the new institute was starting up, early on we had this exposure to various methodologies that are used in EBI, and we were able to sort of get ideas in order to organize our institute. We were also able to collaborate with the Wellcome Trust Sanger Institute in the area of wet lab development, and that was very helpful. So early engagement is extremely helpful and helps shape ideas, helps get new knowledge, and so I think I'm very positive about our engagement in EMBL, and I hope that after today's discussion, we will come up with certain concrete things to do with EMBL laboratories in the area of bioinformatics. So without further ado, let me inform that there are two science-driven talks, so that will showcase, provide you with certain kinds of exemplars of the kind of bioinformatics work that's done, and there are two such 15-minute talks followed by a panel discussion where each speaker gets about 5 minutes, and then the House opens up for a general discussion with the audience. May I now invite Dr. Srinivasan to...
Thank you, Patu. Yeah, I have to use my laptop only for some reason. I have some videos to show. It doesn't work in other computers, so I have to stand here and give my presentation. The cable is too short for me to talk from there. What I'm going to do, before that, I run a research team at the Indian Institute of Science, which has the general objective of understanding the complex relationship between amino acid sequences, three-dimensional structures, functions, and interaction properties of proteins. And we work on diverse specific questions within this broad theme. And my objective in the next few minutes is to highlight three of the themes which I thought is appropriate in the current time in the broad area of bioinformatics or computational biology. The first theme is the marriage between big data and simulation in biomolecular science. So traditionally, intense simulations such as molecular dynamics is usually performed on a single or few proteins. And these days it is possible to do it over a microsecond time scale. But given the advent of the computers and the speed of computers and the way things are moving, it is now, I feel it is possible to combine the big data analysis, that is the bioinformatics, that is the analysis of biological information, and the computer-intensive simulation techniques. And I will just take a few examples on what sort of questions we could ask by combining the two things. The first one, before I go on, I just want to introduce, there are at least three dimensions of complexity in biomolecular systems. This slide shows the complexity in terms of size of the system. You could have the 3D structure of a protein or a protein bound to a small molecule, and then you can have small assemblies. So do you have a pointer, please? Okay. We can have small, like protein-protein complexes. And we can also have large assemblies where the number of proteins or other molecules such as nucleic acids can be bound. There are a number of scenarios in biology where one can think a protein has a role as an individual, and it has a role in a small assembly and also a role as a component of the large assembly. This is one dimension. The second dimension I want to highlight is the functional state of a protein. The 3D structure can depend on what is the functional state of, for example, an enzyme. Here I'm showing, cartoon representation of the kinase, a protein kinase in active and inactive forms. And of course, The third dimension is the time. The atoms are always in motion, and therefore the structure is constantly is subjected to some sort of change over time. So the three-dimensional structure is really an interplay of the size of the system and the functional state of the protein and the time. These three are not independent dimensions, by the way. You can have the connections between the three. So it is 3D in 3D here, which I want to highlight. And I will just take some examples from my own group's work. Here is an example of how the dynamics, the extent and nature of dynamics associated with protein kinases depend on the functional state of the kinase. As you may all know, the protein kinase it phosphorylates the substrates at the hydroxyl group of serine threonine tyrosine side chain, and it can't be doing it all the time, and it can't be kept in the silent mode all the time. That will cause some undesirable effects such as cancers and tumors. So there is a very streamlined protocol for every kinase as to how it can be turned on or turned off, how long it should be turned on, etc. What this picture shows, at the bottom it shows the 1 microsecond simulation of active and inactive forms of a same kinase. And the red shows inactive and blue shows active, and generally, the detailed analysis shows that the extent of dynamics is higher in inactive kinases than active. When you do it for a large number of kinases, this is a big data now, we couldn't afford to do molecular dynamics at the time we did this work, so we have to be contended with the quick and dirty way of the normal mode analysis, the coarse grain analysis. But it is still useful, and it clearly shows that inactive kinases have higher level of dynamics than active. The homologous proteins are known to generally conserve functions, but there is still some diversity when you look at the function at the fine level. And of course, the sequence and 3D structure contribute to the similarity or differences in the function. And we propose here, again taking the example of kinase, that the dynamics plays a big role. Here, in the right top corner, you see number of lines, and the red line in one end of the picture, and then some sort of an yellow or orange line at the top. As you go from red to orange, the extent of evolutionary divergence between kinases increase. And what that picture shows is that closer the kinases are related, similar their dynamics properties are. And the actual place in the three-dimensional structure where the dynamics is similar or different or to do with the binding of other proteins, such as the regulatory domains or the regulatory proteins. And such a difference in fluctuation in correlation can be explained only by the dynamics, not by sequence or structural similarity or dissimilarity. And the allostery is a very important process in the information and transfer in all biomolecular pathways, pretty much. Ruth Rusinov in NIH says every protein is allosteric. And here I want to highlight the significance of this by two examples very quickly. Okay, so here is an example of the cyclophilin, the left top corner, shown in green, bound to the HIV surface protein. It's a host pathogen interaction complex. And the green is the cyclophilin protein when the HIV capsid protein is not bound to it, okay? So, and you can see there's a high similarity in the 3D structure, whether the protein is bound or not. And then the close-up is shown at the left bottom. But, and you can see a remarkable similarity in the 3D structures. However, when you concentrate on the dynamics properties, I can see it here, but I can't see it there. I don't know why. Gosh. Okay, I don't know. Okay. Okay. There in the right-hand side picture, at the bottom, where you see excellent superposition, then the extent and nature of dynamics is different. The vectors which points to the movement of those segments are different depending upon whether it is the bound state or unbound state, and that has implications in the next step in the process. I don't think it's shown here, but somehow it is not shown there. Okay, I have to just show you only the static picture. I'm sorry about that. Here, this is a case of the bovine lactoferrin, which is a two-domain protein, and the crystal structure of a single-domain version and the two-domain version are highly similar in structure. But all that those movies are supposed to show, but the nature of dynamics is different. There are a number of examples where those allosteric sites, which is not always elicited by the difference in 3D structure, also in terms of difference in dynamics, that have implications in something able to bind there or not bind there. Okay, now, the allostery even in small complex systems are shown to be very important to understand. But now we have the advent of elucidation of very large molecular machines, and all the more the allostery plays a major role there. This is the structure of the ribosome bound to the hepatitis C IRES. And the picture in the right is actually a movie, which I'm not going to click. It will become black now, I think. The green are the protein parts, and then blue is the ribosomal RNA, and then the red is the IRAS from HCV. There are important, the information transfer within the complex involving the structural language in communication from one component to another component, which unfortunately I couldn't show you due to technical problem. And the second theme, very quickly, I want to highlight is the bioinformatics aided fitting of atomic models in cryo-EM density maps. I think the afternoon we will have a lot of discussion on cryo-EM importance. What we say here primarily is that from 70s onwards, enormous information on how the molecules recognize each other, for example, protein-protein interactions, has been learned from crystal structures. And what one would expect from cryo-EM structures not very different from those crystal structures. What we are, especially if you have a sort of a modest resolution for a cryo-EM map, for multiple orientations of the atomic models that fitted into the cryo-EM density maps, we can have very similar correlation coefficient. The fitness factor will be highly comparable, but the orientations can be very different. Yeah. I think I have to continue. The time is running out. So what is learned from protein-protein complex structures, crystal structures, is that the interface of the two proteins have residues that are reasonably well conserved. Can you hear me still? Okay. Am I audible without? I'm not audible. Okay. 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 Sorry about this. When two proteins bind to each other, the interface region usually constitutes of residues that are reasonably well conserved in evolution. And when you have multiple possibilities to fit a structure, atomic level model in a modest resolution cryo-EM density map, all that we are suggesting is that try and look for solvent exposed conserved patches, and then try and maximize the occurrence of those patches in the protein-protein interface, and still being sensitive to how good the fitness is in the cryo-EM map. This is one of the many ideas that we thought we can use from the years of learning from structural bioinformatics in the cryo-EM determined structures. And in the similar sort of thought can also be used to validate cryo-EM models, which I don't have time to go into. And another thing is that when we usually fit, let's say a crystal structure in a cryo-EM density map, the crystal structure is determined in its isolated form. And some of the proteins are known to undergo enormous conformational changes. For example, the heat repeat containing proteins. The way one would fit such things in a cryo-EM density map involves an understanding of the dynamics of the system, which are the flexible parts. Again, the right side is a movie actually, which is not unfortunately shown. You can see, you can find the structurally stubborn and structurally very labile regions by doing, the various kinds of simulations computationally, and then again with flexible fitting method, which has a sensitivity to flexible and rigid regions, we can fit the models into cryo-EM density maps. And the final part is something to do with sequence analysis. Especially I think the EBA colleagues will be interested to know this. Once the genomic data is, sequence data is deciphered, you have the sequences of proteins, and the first question is, what are the functions of these proteins, structures of these proteins? And we have a lot of sophisticated sequence search methods that will tell you the relationship of these proteins to other proteins that are already studied in the laboratory. However, large number of proteins still escape from all these sensitive detection methods, and it is not really the problem of the method here. It is more the problem of distribution of sequences in the sequence space. If you imagine the sequence space something like this, where every blue encompassment corresponds to proteins of the same fold, and then all yellow compartments are the homologous protein families, closely related protein sequences. And many methods depend on the availability of such intermediately related sequences in order to traverse using the sequence search methods from one family to another family. So let us say your starting sequence, sequence for which you want to detect the homologs, is somewhere close to this family. And then because of the void and sparse region in the sequence space, whatever is the sophistication of the program, you can't hop from here to here. So what we decided to do some years ago is to generate protein-like artificial sequences using computational methods and fill in those void and sparse regions so that the facile travel in the sequence space is now possible with any of the sequence search methods. We have done a number of things with it, and one of them I'm showing you in this slide, which says that for more than 600, now 700, 800 families, now we keep doing it, of domains of unknown function, we are able to find the relatives of known function and structure. I think this is just an elaboration of, just to make sure that once you have the distant relationship detected, which cannot be detected by if you don't use those special sequences. We just embed these artificial sequences into any sequence database of our choice, and then we run the sequence search program of our choice. Any sequence search program would do. The real trick, the real difference is made not by the search program, by these sequences which are artificial in nature. So I think I should stop here. Yeah, sorry, I took a little bit more time than I should. So I basically emphasized 3 themes in which the computation is playing a big role, and then especially the first and third themes involve integration of the high volume of data and the simulation techniques. So I'll stop here. To recognize the functions of proteins encoded in the genes. And all that you are saying is that the detection of related proteins is probably the typical first step in order to give ourselves an opportunity to say about the function of the protein. Even now, even with the today's set of lots of HMM-ER sort of programs, if you run any of those programs for the entire set of proteins in any genome, especially the protozoan organisms, large number of proteins, you don't have any relatives detected at all. It doesn't mean that they are all new proteins. Nature have not explored the relatives of those before. There could be a small proportion. Usually in the retrospective analysis, I kick myself. Oh, this is already there, but I couldn't detect it before. So all that I'm saying is that it is not always the problem of the program. It is the problem of the database sometimes. Despite so many sequences that are piled into the sequence database, you have some sparse or void regions breaking the continuum of the sequence space. So you have proteins in the same fold, for example, but not necessarily all of them had to be, say, homologous. But there could be, due to convergent or divergent evolution, there could be a sequence continuum. When the continuum is not represented in the natural sequences, all that we are saying is that taking the help of the structure and previously detected evolutionary information, we just fill in those void and sparse regions by protein-like artificial sequences, which are generated by considering the distribution of sequences in 2 families within which the natural intermediates are not available. So we fill in with artificial intermediates. So in order to use this, what we do is we have these sequences which are generated. None of them is really natural, but they are all connectors of natural sequences. We just integrate them into you might have your own proprietary database, or you might have some, anything, SwissProt. You can integrate it in any database you like, and then you can do your sequence search. So these sequences could help the, the traversing of the sequence search program in the sequence space, and thereby enabling the distant relationship detections. I can show you a number of examples where the query is, nothing is known about it, for example. The structure of it is not known. And there are lots of hits of natural sequences. Nothing has known structure. But during the SciBLAST search, for example, it has traversed through these artificial sequences. Every artificial sequence has parents, so it has the notional fold. So therefore, the query and hits, we can associate the fold to it. I don't know whether I answered your question, but we can talk further later.
Good morning, everybody. So just now I was thinking actually how I am related with the EMBL actually. I am associated with the EMBL directly or indirectly from last more than 25 years. Because in our institute, my responsibility is where I was working earlier to maintain the email. Okay, it was a big thing if you go 30 years back. So I was providing service to the institute. And normally I access the softwares and databases from EMBL. How I access them? We send the email, and from email we got the software from the EMBL actually. That's the first time I got the exposure with the EMBL. I thought lot actually. Because in India I have not seen that type of culture that all the softwares, including their source code, databases were freely available to the public basically. I thought, why they are providing free to the public? I was really impressed actually. And I decided at that time that if I come in the science, if I develop anything, it will be free to the public. And since 91, I started my own career, developing the softwares, databases, and all software, databases, dataset, whatever we have developed in the group, they are all free to the public. So this was the first interaction with the EMBL, and most of the data I have taken from the NCBI, EMBL, and these resources. Okay, so I thought it's a good job they are doing from a long time, whether people are upsetting or not upsetting, but EMBL have done the excellent job in last number of years basically. This is my feeling basically. Though directly I related with the EBI, I worked for some time in EBI Cambridge for some time actually. So today, I'm not going to talk about my work, what I'm doing actually. Recently, what I did actually, I compile most of the softwares and databases developed in India for health point of view, how they can help in health informatics basically. Though they are not complete, they are basically just for the health point of view basically. Today, I'm presenting that portal basically, and same time, Dr. Debashish Mohanty from NI compiled all the databases this year itself, all the databases developed by India in last number of years basically. Okay. So this is the address of the our portal. So this table I have borrowed from Debashish Mohanty's paper recently published on this one. It shows the position of India. They have collected, compiled all the papers related to databases, biological databases, and then they have ranked it. So India have contributed quite well actually in the databases, and we have contributed quite reasonably. And right side, I have given the statistics of portal we compiled. So what we have done actually, all the servers, 212 web servers developed by our group, they may be softwares, databases, and datasets. They are freely available. They are online, and I have provided link to the 300 other Indian servers, basically. This is not still complete. I'm still committing that this is not still complete, but all these 500, they are active, they are working. That's a major challenge even for the EMBL in future. How to maintain these databases, services for future, because a lot of money is required, infrastructure is required for that point of view. So there is a compilation of servers, all abstract content, and limitation that still it's not containing all servers. So we divide into the different headings. Let's say genomics. We maintain all the genomics related informations, all the servers which is related to the genomics related information that include annotation of genomes, cancer genomics, genome medicine, genome biomarkers. So I'm not going in the detail of each and everything. But we have compiled all the information. So here Professor Majumdar says that in India, though structural biology is dominant, but fortunately from last few years, maybe let's say last 15 years, number of people have come in India where they are working in the different area other than the structural. That's a good part. So I'm showing you the Indian servers. I have divided into different area, and in each section, sufficient number of web servers and databases have been developed. Though my aim is not to focus on India only, in initial phase, we are only compiling for the Indian resources which can be used directly or indirectly for the health informatics, healthcare. Later on, we are planning to compile all over the world database and these resources that for the health informatics, because the ultimate goal of bioinformatics, according to me, is to serve the public basically. So there is a different sections. I'm not going again in each section. I will not take much time. Yes, this is the objectives our group have in mind for the future point of view. It's very clear: computers and resources for newly emerged diseases. Today we are getting the different diseases, different viral diseases, bacterial diseases. The question is, despite we have a lot of servers and databases, can we handle these diseases effectively in time manner basically? Experts are there. They are maybe in the EMBL, they may be in some other institute in India and other places. But the health persons who are working in the health or who are in the biologists, it's very difficult for them to take the new genome, analyze it, to find out the targets, to design the drugs. It takes a lot of time. Can we have some system where we got the new virus or bacteria, sequence it, because it's very quick nowadays, and just propose that these are the possible drug targets, biomarkers, and vaccines. And the biologists will use directly them. So that will be the service to the community directly. So that's one topic we are interested. Another is basically informatics protocol, health-related problems, means what type of problems we have in the health and how we provide a solutions to them. Some are not related with the genomics, they are different type of problems. Another is genome-based solution for personalized therapy. That's very interesting phenomenon. Though we are talking a lot that personalized genomics, but still, it's not in real life actually. Lot of data we are generating a lot of data. Still, there is a lot of gap between the ground reality and data generation. Okay, so that's another challenge. Another is strain-specific prioritization of drugs basically. Let's say HIV, it was a problem if you go 30 years back, or, because it was changing quite fast. Now we understand that for the mutated HIV, we can generate the drugs fast actually. Okay, so that's the type of problem should be there. Genome card, we should understand drug toxicity and immunogenicity like that. So here, now question is what we expect from the EML and what type of collaborations, let's say, we want actually. For, as Professor Majumdar say, whatever we say, still for the big data, like next generation sequencing data and other, we are still behind to the, let's say, 5-10 years than the EMBL and other organizations. So if we have the better collaborations with the EMBL type of organization or NCBI, and we are also having a lot of expertise, computational power, developing the web server capabilities, so we may work better basically. That's my theme of this talk. Okay, thank you very much.
Sorry. Okay, I will try the same trick to speak from here so that I can see what's coming up on my slide deck. Otherwise, if I do it from there, it will feel to me like a PowerPoint karaoke, so that I don't know what is the next slide, even if I have created them myself. So first, thank you very much for inviting us here to present some of our work, and I hope that looking how much discussion we had yesterday, I'm pretty sure that we come again to a lot of areas where we could help each other. So I will speak a bit about the EBI's work on bioinformatics research, and especially on services. But I should also say that EBI is not the only place at EMBL where bioinformatics is happening. There's also very strong there are also very strong groups in Heidelberg doing bioinformatics, and also there are bioinformatics activities around other areas of research at the different sites of EMBL. So before I speak about EBI, let's look a little bit back and be proud of our achievements in the life sciences. Because biology is now the leading scientific discipline. If we have some high energy physicists here, sorry about you. You had your century, now it's our century. And we have a real shift going on, that biology was in the past small scale and mainly hypothesis-driven. It's still a lot of hypothesis-driven research that will not go away, but we are now in an era where we have a lot of data-driven research, which also means we need to have bigger groups. And especially the generation of sequencing data and other omics data is driving this process, and the Human Genome Project was in some way the big bang event in biology. Oh, sorry, you don't see that, but there's something popping up here that I want to get rid of. So this picture here shows the growth of data in the EBI databases. This is a log scale, yeah? It's done by different archives where we store different data. So the red line, which is on top there, is the data available in the European Nucleotide Archive. So that is the EBI's part of EMBL GenBank DDBJ, where we store all the sequencing data which are completely and publicly and openly accessible. And you can see that there were some very steep, there was quite a steep curve for a while. That was the beginning of next-gen sequencing. It was a bit of a problem for a while for us. It doubled every 6 to 8 months, but with better compression algorithms, we got that sort of under control. Now it's back to the normal growth rate of doubling every 18 months. The green one is an interesting one. That's a database we started much later, and that's the EGA, the European Genome Archive, and that contains data on humans which is controlled access. That means these are humans which could be identifiable, there's phenotypic information available to it, and that is only as accessible when you have approval for your analysis by a data access committee. So, as you can see, human data is now incredibly, controlled access to human data is now an incredible amount of data. There are around 500,000 complete genotypes or genotype, 500,000 sequences or genotyping of 500,000 complete human genomes. So And that's a very, as you can see, it's following the curve of overall accessible data. The brown line there in the middle, that is Array Express, you can see it's still growing, but arrays using arrays in science is a sort of dying art. People have still the machines, their equipment, so they use it still, but it will at some stage go out of business and will be replaced by RNA sequencing completely. The sparsely dotted line, I don't know what happened there. Some of the pixels got lost from here to there. That's proteomics data. What you can't see, it has actually overtaken the array data, but as I said, the pixels got somewhere lost in translation. And the line down, the last line is the metabolomics data in the metabolites database. You can see all of this is still growing, is growing very rapidly, but sequencing data is really still the very much dominating factor. So that's where really EBI comes into play. Because we, at EBI, we try to deal with all this type of data. We are on the Wellcome and Trust Genome Campus, together with the Sanger Institute and a few other players. At EBI, we are around 600 members of staff, and we also host something which is called the Alexia Technical Hub, and I will say a few words about that later on too. So I should mention that we, of course, get most of our money from EMBL directly from the EMBL member states, but around half of the money comes from other funding agencies, the European Commission, the Research Councils of UK, the National Institutes of Health, the Wellcome Trust, and from industrial contribution. Why did I bring this slide up? Because there's one question, at least in Europe, I always get asked about that. What about Brexit? So we are part of EMBL. We are an integral part of EMBL, and therefore we are not part of a European Union structure. We are an intergovernmental organization on our own. The UK was a founding member of EMBL, was always a strong supporter of EMBL, and we have no doubt that this will be also in future. So we have a host site agreement with the UK. We can hire whomever we want from wherever in the world, and this will not change. We will be still eligible to apply for funding from the UK research councils, and as part of EMBL, we of course can also still apply for funds from the European Union. So maybe this was not a question you wanted to raise, but usually it comes up. So it saves us time later on in the discussion. Our mission at EBI mirrors the overall mission of EMBL. Of course, we want to deliver excellent research, but our main mission is really delivering scientific services, train the next generation of scientists, and you hear later on more about that from Kev, to engage with the European industry and coordinate as part of Alexia bioinformatics research infrastructure in Europe. So in our service mission, we really want to enable life science research and its translation into various areas of life sciences. What does that mean? Well, what usually happens is labs around the world send us their data, we archive it, we classify it, we share it with other data providers, then we analyze, add value and integrate it, and provide tools to help researchers to use it. And then hopefully, the users make good use of it, get new clever ideas, design new experiments, create new data, and send it to us. So it's a collaborative cycle. When we moved from EMBL Heidelberg to EBI, to Hingston 23 years ago to set up the EBI, there were only two projects existing. That was the Ambl nucleotide sequence database, and that was SwissProt. That was all the projects we were involved in. Since then, a lot of other activities came up, we put them into so-called clusters of related activities like genes, genomes, and variation, with Ensemble, for example, gene, protein, metabolite expression, like with databases like PRIDE for proteomics, the expression atlas, and so on, protein sequences, families and motifs, Interpro, PFAM, UniProt, molecular structures, our contribution to the PDB in Europe, chemical biology, systems biology, a lot of molecular archives, literature and ontologies. So, and one thing which is striking, that all of these major activities are done in international collaborations already. But you see, here is still a lot of necessity to bring some of these dots over here. So we hope for that. And I know that Samir Velanka was here just in August to talk about partnering between India and the PDB and to set up long-term PDB India here. And I think that would be a great first dot to put on this map for the future. So we are not only collaborating globally, we are also have a global community. We serve a global community. And you can see there are a lot of dots also from India. Around 10% of our usage comes from India. So it's significant. It's quite significant. Overall, we have every day around 27 million hits against our websites. Per month, around 5 million different IP addresses are logged into our databases. And we run millions of computational jobs for external users on our websites. To do that, we have around, right now, around 200 petabytes of spinning disks in our data centers and around 50,000 cores. And we expect that in 2020, we will hit the exabyte age and needs around 200 to 250,000 cores to do our job. So, a few words about the research activities we do. Our campus is really a very, very unique setting for research because we have a super rich technical environment and we have one of the world's highest concentrations of expertise in genomics and big data in biology. So our research and service activities are mutually supportive and we have a lot of people who have dual roles and so it's really, we exploit that quite nicely. That are the current research groups at EBI. These little symbols show the different scientific background they have. So as you can see, it's not only life scientists and chemists and physicists and medical doctors and everything. There are, as I said, some people like Pedro Beltrao or Anton Enright, they are full-time researchers, and people like Alex Bateman or Ewan Burney, the other director of the EBI, Alvis Brasma, they have a small research component, but their main role is on the service provision. So the workhorses, of course, are always the postdocs and PhDs. They do the real work, and we have special projects to foster interdisciplinarity, the so-called iPods across EMBL, and we cloned this system on the campus to have joint fellowships with the Sanger Institute and on the Cambridge with the Cambridge Clinical School to have more biomedical related computational projects together with the clinical school in Cambridge. And I will not say more about training, because Kev will talk about that later on. And yesterday it was already mentioned, the EMBL International PhD program, since Ian talked about that already, I will not go into detail about that. But we recruit, of course, in the framework of EMBL in the same way. So for us, also important is industry engagement. So we have a long-standing tradition of supporting industry at EBI. Our industry program is more than 20 years old, and we welcome interactions with industry through pre-competitive collaboration, standard development, technical development, and service development. What we are not doing, is contract work. It always needs to be in collaborative, pre-competitive, and open data sense. These are our current industry program members. And what we do for them is mainly running member-selected industry workshops. So it's training for industry in some, you can say. And these are some recent examples of courses we did for our industry partners. Since bioinformatics gets more and more translational, we also moved towards doing more bigger projects with industry. The first one was in the area of drug discovery. Finding the right biological target for a drug requires bioinformatics to identify promising targets and select candidate medicines, and we have various databases which are very helpful in that. So a while ago, 3 years ago, We started the Open Targets project to pinpoint the process in the human body that have a demonstrable effect on disease, and that should help to improve the success rate in the discovery and purposing of medicine. What we do in this project, it's together with the Sanger Institute, with GSK, with Biogen, and there's a third pharmaceutical company in the process of signing up to, is to build a huge matrix of all diseases and all human genes and say what's the evidence that this gene is involved in this disease. And we also commission then a lot of additional experiments on genome scale at the Sanger Institute. To give you a bit of an idea of the scale of the project, there are around 70 FTEs working for the next 10 years on this project. So it's a substantial private-public partnership. We are a nice big place already, but there's a lot to do, and we need to always to do that in an international context. In Europe, the vehicle for that is Alexia. And Alexia has in principle the same mission as the service mission of the EBI, but on a European scale. These are the current member states, the Alexia Hub, which is situated at EBI, is trying to build the glue between all of these activities so that we don't duplicate things, but really work synergistic together. And I hope that you get now a little bit of a flavor of it, and I guess you get also some ideas where India will fit in future into this framework. With that, thank you very much. I will hand over to Kev.
Good morning, everyone. Can you hear me? Loud and clear. Good. Okay. Well, first of all, I'd just like to say what a delight it is to be here. I feel really privileged to have been invited to this event. Thank you very much. So I'm going to talk to you a little bit about the EBI training program, and just to make sure that we're clear on the distinction between what Rolf has already touched on, the training of our own PhD students and postdocs. The focus of the program that I'm going to talk about is entirely external. So what I'm going to be discussing is how we engage with our users and potential users around the world and ensure that they can make the most of the biological data resources and tools that we provide at the EBI. You've already seen this slide, but I really just wanted to emphasize that training is at the heart of the EBI's mission, and as an organization, we view it in that light. The entire organization views training as a core part of what we do. And without that, it wouldn't be possible for me and my team to do our job. So why is bioinformatics training so important? Well, I don't need to tell this audience that biology is increasingly data-driven. Whoops. I've been caught by the auto-advance bug here. We touched on this a little bit yesterday, but bioinformatics is not only important for curiosity-driven research so that we can understand the biological world around us, but it's also becoming increasingly applied to medicine, to agriculture, secure and sustainable food production, and to the environmental sciences. And this means that we have an ever-growing constituency of users of the EBI data resources and tools, many of whom have never had any formal training in bioinformatics whatsoever. And so this is the core part of what we do in the training team and the training program at the EBI. And essentially we have two jobs to do here in order to ensure that tomorrow's biologists look more like today's computational biologists, because big data really is at the heart of what we're now doing in biology. So the bit that we focus on at the EBI, and I think the bit that we can support you in the most, is enabling the scientists of today to get up to speed in terms of bioinformatics. Of course, there's another really massive job to do, which is the next generation, inspiring and enabling the next generation to become, if you like, compute native right from the beginning. This is something that I think is way beyond the capability of the EBI as an organization, and it's really up to the universities to grasp this problem and ensure that something is done about it in the near future. I have a few ideas about how the two can interact, but I wouldn't say that this was our core activity. So how do we actually go about training our users? There are really 4 main strands to our activities. Our training program is now 10 years old. We've just had our 10th birthday. And at the heart of our program is a series of on-site courses that we run in beautiful state-of-the-art training rooms at the EBI. This is rather... It's probably the most advanced training that we do, actually, and getting onto these courses is highly competitive. We have a very transparent selection procedure. We have far more applicants for our courses than we can actually fit on the courses. But applications are open to anybody around the world. And in fact, last year, we trained about 400 people through the program. Through this part of the program, many more actually through the program as a whole, and about 23 of those people came from India. So we are already serving Indian science in this respect. But of course, it would be great to see more applicants from India. We also go around the world, and in fact, this is the largest part of our training program. It accounts for by far the largest number of person hours of training delivered in any one year. And a really lovely example of this is the 2 workshops that we ran back in 2013. One in Kalyani, hosted by Pata, and one in Bangalore. And I'll say a little bit about those later on. But by far the fastest growing area of our training program is Train Online. Now, these are freely available, openly accessible courses, mostly tutorial-based, mostly aimed at enabling people to make the most of our data resources and tools. And typically they're around 2 to 3 hours long. We have a number of shorter courses in Train Online that focus more on bioinformatics concepts, where the data come from, the things you have to look out for when you're analysing the data. And then we have a number of video-based courses that are loosely based on some of our face-to-face courses, but presented in a rather different way. The whole concept behind Train Online is that it's kind of instant, on-demand access to EBI training. I already mentioned that our face-to-face training program is highly competitive, and we really wanted to do something for all of those other users who couldn't make it to an EBI course. Industry is also a very large user of Train Online, and in fact, if we look at the geographical distribution of users in Train Online, India is our second largest user. Recently, over the last 2 years, we've added webinars to our online training offering. These are short, live presentations with question and answer sessions. We offer them on a fortnightly basis every Wednesday afternoon, and then we make those pre-recorded webinars available through Train Online, so there's an archive of them that can be watched at your own leisure after the events themselves. And the auto-advance bug has caught me yet again. Finally, in terms of building capacity in training, 4 or 5 years ago, we started a sort of trainer support activity. This started out very small. We were working with our collaborators in Australia as part of their new associate membership of EMBL. And it's actually been tremendously successful. So every year they've been sending a delegation of somewhere between 5 and 8 bioinformaticians from all over Australia. They aim to get a spread across all of the different state capitals. And they spend about a week with us. We do some basic kind of andragogy 101. The training that we do involves having people from a large number of different backgrounds with very different experiences in the room at the same time. And for anybody who does that, that can create a number of challenges, actually. You have to meet the expectations of all your trainees, and you have to ensure that everybody gets what they wanted out of the course. And so over the years, we've developed a number of tips and tricks for ensuring that we do that. And really all we're doing is passing on what we have learned. They then get to experience an on-site course in the flesh. They participate in that course. And then afterwards, we sit down with them and we work with them to develop their own course programs, which they can then work towards delivering back at home. So we've done this at about 5 or 6 different topics over the past few years, and this really has had an impact in terms of bringing the bioinformatics training community together in Australia. So I mentioned that it was our 10th birthday recently, and so it's been quite a reflective time for us, and we've been thinking about, well, you know, what has made the program work? What makes it tick? And I thought it would be useful for me to bring a few of these things up so that we can think about what might help to stimulate a strong bioinformatics training community in India. So the first thing is that training really is an EBI-wide effort. So Rolf mentioned that we have about 600 members of staff. I know that at least 165 of those people were actively involved in our training program last year. So that's the kind of proportion of people who are really actively involved in either developing training materials or delivering face-to-face training. And one of the reasons why I know this is because training at the EBI is now centrally supported. So there is a core piece of budget that any member of EBI staff can charge their time to if they are involved in developing or delivering training. And so I think this has made the entire job an awful lot easier, actually, because people can see that training is well-resourced within the organisation, and it's not competing with their other activities in the same way as it might anywhere else. All of our trainers, or the vast majority of our trainers anyway, have another full-time job. They're either biocurators or software engineers, software developers. And so, you know, there is a risk that delivering training could compete with those other activities, and we wanted to do something about that. And I think that's been, well, it's been a massive boon for the training program. So the point I really wanted to make there is that if you're going to do training, you really do have to resource it properly, and not just in terms of money for teas and coffees and travel, money for the time that is put into developing the training materials and delivering the courses. I think our values, which we established right at the beginning of the program, have been tremendously important to its success. We believe in immersive training, bringing people together in a collegial atmosphere. Our training is led by the experts who are developing and adding content to the data resources and developing the tools. That enables our training to be cutting edge because it's the people who are working on the next thing who are delivering the training. And we also put a huge amount of effort into being responsive to our trainees' needs. We do the usual post-course surveys. We find out about our trainees before they actually come to us. And we're also doing long-term feedback surveys, 6 months after each course and up to 2 years after each course, so that we can really find out what impact our training has had in terms of the research that people are doing, in terms of the collaborations that they've struck up, perhaps the papers or the theses that they've published. And of course, we have a high proportion of practical content. We want people in an in silico sense to get their hands dirty and to make use of the data resources and tools when they're training with us. I think another key to our success has been our philosophy of openness and our desire to collaborate. We make all of our training materials openly available through a Creative Commons license, and we actively encourage others to make use of those materials, to tweak them, to make them fit for purpose for their own needs. We make all of our materials available on our own website, and we also share them through aggregators of training information. And the Elixir Training e-support system is one such portal where you can go and find information on both our courses and access to our training materials. And I think we really thrive on collaboration. And I've just got a few of the large consortia and professional bodies that we collaborate with listed here. I mentioned yesterday that we've got involved in training managers and leaders of research infrastructure. So I just wanted to highlight the RI Train project, which is the consortium behind those activities. But most of the rest of what you see there is much more intensely focused on big data and bioinformatics. But these collaborations have enabled us to contribute across the globe to a number of different things. We've put together learning frameworks that can be used by anybody training in bioinformatics. We've helped to build new training programs, and we've shared best practice. I just wanted to say a couple of words about the 2 workshops that we ran in India, because I think they're a beautiful example of how we like to collaborate. Parter actually set us quite a challenge when he first contacted us with an inquiry about delivering some training in Kalyani, because the vast majority of the off-site training that we do is quite tightly focused on our data resources and tools. But what he was looking for was a course in NGS data analysis, and he was particularly keen that we provided some training on RNA-seq and ChIP-seq data analysis, because he already had the capability to train on genomic, whole genome NGS analysis. And so we had to put together a rather different off-site workshop, but this became a really excellent collaboration between our 2 organizations, and the training was delivered jointly, which is not typically the case when we run an off-site workshop. Usually it's us doing all the training. And Parter also identified some kind of would-be trainers for the future who our trainers worked with while we were in Kalyani, and that has enabled them to go on and deliver further training courses. And I think this is the kind of collaboration that I would really like to see more of with India in the future. Final word about what's made us successful before the mic pegs out on me is our scalability. And here, again, I wanted to highlight the power of Train Online, because face-to-face, we typically reach about somewhere between 10 and 12,000 people a year. And this is through our kind of formal classical training courses, but also a large number of scientific engagement activities that we get involved in. We're invited to deliver training as part of many other programs, in addition to running our own programs. And we also go out to conferences. We do workshops at conferences. We do conference exhibitions. So there's and of course there's all the knowledge exchange activities that we run with industry as well. So that's taking all of those things into account, and we can still only reach around 10,000 people a year. But through Train Online, we really can upscale. And last year, over 400,000 unique IP addresses accessed Train Online. So it's a completely different scale. It's also, as I mentioned yesterday, a completely different type of training. And I'm not trying to suggest that one replaces the other, but it does at least make high-quality bioinformatics training available to a much, much larger audience. And the same goes of the webinars. And so all of these things really are contributing to the vision of my team and the program as a whole, which is to deliver a program that enables scientists at all levels, regardless of their location, regardless of their sector, and regardless of their career stage, to make full use of public biodata and to share their experience and their expertise openly with others. And with that, I would thank you for listening. I'd like to introduce you to my team. This is actually a slightly out-of-date slide. The latest addition to my team is from New Delhi, and he's doing all of our web development for us. And unfortunately, I don't have a photograph of him yet because he joined so recently. If I can, I would like to make these slides available afterwards so that you have all of the URLs and all of the information you need to access different parts of the training program. And of course, I would also like to thank our many funders. The latest funding that we received was from the Global Challenges Research Fund. We've got a nice chunk of money to do some really, really interesting capacity building in Latin America. and perhaps there's potential to apply for this kind of funding in India in the future. Okay, with that, I'd like to thank you for your attention.
I don't know yet who, but
from the present, it's working. From the presentation, you said that there are 23 Indians.
who of us?
Okay. 23 Indians are all applied for training in EBI last year.
So I think it was a larger number who applied. We had 23 Indian nationals on the program as a whole. Yeah.
And you also do workshops in India with our NIBMG team, with the CCAM team. So my question is, given that some activities are already being carried out in bioinformatics and Indians have access to it, how then does becoming an associate member enhance? And this is something which we need to understand because a lot of investment and what in order, we have to think bigger and larger and get our returns on investment back.
Yeah.
And that the panel needs to discuss, and bioinformatics people need to let us know how it works.
Yeah. Yeah. So
I need your inputs on that.
So, the training program has always been accessible to anyone from around the world, but we pay special attention to our member states. So, we want to ensure that we can now make a coordinated approach to first of all perhaps make it clearer how to apply successfully to our on-site training courses. So there's a piece of PR that we can do around that, and just ongoing conversations I think could enable that. And then also I think your membership of EMBL gives us an opportunity to have a much more coordinated approach to delivering training here in India. So, for the first time, this has really given us an opportunity to get those who have an interest in bioinformatics together and to think about, well, what can we do? Where are the real foci of interest, and how can we help to strengthen capacity in those areas? So it enables us to, I think, work in a much more engaged and collaborative way, rather than in the rather ad hoc way that we've worked in the past.
So, Kath, can I make a greedy proposal? When we become an associate member of EMBL, is it possible for India to say, you announce your workshops ahead of time, so we know what workshops are going on in which days. Let's say that we from India somehow, through a selection process, we identify 5 or 10 people. Could you reserve 5 or 10 seats for us as an associate member state in the workshop?
No, we don't reserve places. We have to have a completely open process, and that's true for all, regardless of whether someone's from a member state or not from a member state. We have to make that a completely open process. But I think there's information about our selection process actually published on the website. So there's nothing magical about this, but actually just pointing people to that and giving them some tips and tricks as to what we're looking for. There's no rocket science involved in this. What we are looking for are people who are going to benefit from the training. We're not looking for those people who have the longest and shiniest CVs. We're looking for those people who might perhaps have just started out in analysing a particular type of data. They already have data in their hands, or they're about to have data in their hands. They desperately need to do something with it. And so it's kind of, there's quite an art to picking people who are at that sweet spot where they're really going to benefit most from the training.
Well, Kev said already a lot about that, but of course there will be, we hope that this will lead also to the additional ability of us to provide more. Yeah? The details are not clear to me right now how this will all work. So we need to be a bit careful with expectation management, because we are not in the know of all the details. But of course we hope that if there is additional financial input going into EMBL, that this leads also to us being able to enhance our capabilities. But Kev said that very careful. Until all of this is sorted out, we don't want to promise what we later on can't step up to. Yeah? So it's a process which is under development, and it's also that we will definitely not go ahead and say each member state or associate member states gets a quota according to the financial contributions. I think this is just not how it works. That would be disastrous for EMBL as a whole.
Hello. It's very impressive to know from the MBL side that there is a lot of effort on the services and on the training in particular. I was wondering whether you also participate in joint PhD programs wherein students who are registered in Indian institutions can also get a dual degree by working in EMBL for a substantial time. Perhaps.
Yeah, the answer is in principle yes, but they always need to go through the EMBL PhD program process. Then the details how, I don't know exactly how many of the students, Indian students we have are right now also in an Indian university. So all the ones we take are enrolled in Cambridge, for example. So they are not enrolled anymore in India, but the Indians are then enrolled in Cambridge. We have an incredible amount of applicants from India, and there we have right now a problem that we can't evaluate necessarily in the best possible way the potential of the students. This would be for us a real improvement if we have a partnering side on the other side who can do a pre-screening and say, look, we understand their CVs, we understand their home institutions, we understand how good they are on paper. These are the ones worth for you looking at. At the moment we get 1,000 applicants per year for 80 positions overall or something like that. So it's humongous, and I don't think that we do very good judgment to these applications. So that would be an incredible step forward for us.
My question is to both Raghav and Srinivasan together.
Thank you. Just to add to this, once we have the membership, associate membership in place, we also have a possibility to have a collaboration with an Indian institution, academic institution, for the joint award of PhD degrees. So this is something that will add to what Rolf already mentioned, and will really help the selection of participants. Just to add to this.
Yeah. So, see, what we saw is, though in India we have several databases or web resources developed, they are not closely integrated with primary data repositories, unlike EBI, where the primary data repositories as well as resources are under one roof. So what we generally find it difficult is to keep these resources updated and all. So do you think in India we should try to integrate both? So bioinformaticians should also be involved in developing data repositories in consortium with the EBI or in not only structural biology or any other areas.
No, that's absolutely true, and there are definitely there's definitely a lot of ways how to improve it. At the moment, we have, for example, I gave the example of Samir Vilanka before. There's an EBI PDB India partnering award by BBSRC, where we try to work out in this field something, and I guess that needs some. If we come to this stage, we need to have some sort of strategic discussion how and we can link that better, and what would be the funding vehicles, whether this would be through partnering labs, that there would be special partnering institutes, partnering labs in India, which would provide the interface to us. Because we can't interface with all the institutions which are out there in India. It's just impossible. So we need to be selective, but it should be selective based on excellence in India and what it can contribute to open science in the world. And that is something where we need your help, because you have the insight of your strengths and weaknesses, and when we can have a common selection process how to do that. So I think we are just at the beginning of the road, but as long as we have a clear shared objective and vision that we want to bring up the work which is in India into the visibility of the whole world and link it with the rest of the activities around open science databases, then I think we're on the right path.
Hello.
It's working, Rawa, it's working.
This is an important point. In India, we started from distributed information centers from BitsNet. Individually, we are working fine, individual groups are working fine, but unfortunately, in the last number of years, despite the success of bioinformatics in India, we do not have any formalized way of some center or institute where the infrastructure like EBI should be here. So what happened, we are developing software databases, they are not maintained for a long time, basically. That's a major part of the problems. If we are dependent on the grants, if we got the grants, then we are maintaining for some time and then it's not working. So it's important for India, if we want to sustain that, similar to like NCBI or EMBL, some center or institute should be there where the funding should be there for the long term, basically. And we can maintain the infrastructure. Infrastructure is the most important part in bioinformatics, basically.
Just to dampen a bit your enthusiasm, yeah? So, as I showed before, half of the money for the EBI services, more than half of the money for the EBI services comes from grants which are 3 to 5 years long. So we have institutional funding from EMBL, which we use for our hardware and other infrastructure in the backbone, and for the basic maintenance of what we see as our core contributions into the cosmos of life science databases. All development work, or nearly all development work for, adapting to future technologies, future scientific trends, comes from third-party money. And to be honest, I also don't think that this is, sometimes it's a bit hard, but it has two advantages. First, if you need to convince your peer reviews that this is a worthwhile investment to go into this area, then you have a stamp of approval, and this is a good thing. The other thing is also it forces us to do a lot of collaborative grants. Nearly all of the work we're done in collaboration, and if you do collaborative grants with researchers, then you know where the field is going, and you get some real-world problems. Databases in certain areas which were shielded from competition and cooperation sometimes got very, well, difficult to use and adapt to the trends in science. So I think I think it's good to have a stable anchor funding like we have, and I would of course prefer if we would have a bigger share of EMBL money to have even more stability. But the principle of having a mixture between institutional funding, some anchor funders who give where we can always go to, and some more ad hoc science-driven funding is a good mixture. And I wouldn't like to throw that out, because then we would lose some of our strengths.
When you're talking about collaborative research grants, once India becomes an associate member, it is possible for Indian scientists and EMBL scientists to develop research, collaborative research grants, and apply for EMBL funds. Is that right?
I don't know, to be honest, because I'm not involved in the discussions how the partnership should work later on in detail. So taken from the other, looking into the other partnerships, associate members we have already in Australia and in Argentina, it doesn't work this way. You're not applying for EMBL funds, but there's EMBL money we use for interacting with the partner institutes, and when the partner institutes have their own internal funding to interact with us. What we can do is then to go funding agencies like the Wellcome Trust, like BBSRC, who have special, the Newton Fund, for example, who have special funding streams to foster such interactions. And if you have already an established collaboration and know what you're doing, then you of course have a higher chance of success in getting such a grant. But it's a third-party grant, and it's not EMBL money. So as I said, I don't know the details. I'm not Ian, but I would guess, very strongly guess, that this will work in future, similar to the situation with Argentina and Australia. And so I think my answer is probably correct.
Given that EMBL funds will not be available to Indian scientists in a collaborative research, so is it possible, and this maybe Plamina can answer and maybe all of you can answer, is it possible that it is co-funded, we have collaborative research grants which EMBL funds the EMBL scientists and DBT or any funding agency within India funds the Indian collaborator?
So this is something that, as you know, we have been discussing, and the answer to this is very close to what Rolf already given, that when we engage in collaboration, we are, of course, planning to engage various collaborations, then from our side, we'll use the EMBL resources to support these collaborations. So it won't really be funding a project because we're not a funding agency. This is something that EMBO, for example, does, so there are other funders. But when we engage in this collaboration, it will be co-supported by EMBO, obviously through EMBO's funds for this.
And EMBO resources, in facilities and resources.
Exactly, yes, correct.
That's not working.
Yeah, I just want to share my viewpoint regarding the keeping the web resources and databases updated and keep going. What I understand, in the Western world, when you have a popular database, people apply for grants, and if they get funding, they could employ exclusive staff and postdocs or some people whose full-time work is to work on the maintenance and update of the databases. I think I'm right in my belief. But my experience here in this country is that the main workhorse here are the PhD students. And when a student develops a resource or a database, especially the database, and he or she writes a thesis and goes away, there are two challenges now to keep the database going. Number one, to keep it updated. If you don't update it, there's no use. And then the student who is going to update, it doesn't make that update work. doesn't make an important component of his or her PhD thesis. So the student feels not a worthwhile thing to just update it. So we have the problem of not only just the capable student doing on the work, and the main mandate of the student obviously is the PhD thesis. How an update of a database, adding a few more structures in a structural database and organizing it would make a chapter in the thesis. This is the problem here.
So again, I mean, this is getting a little bit tangential to the core discussion here. I think we need to have what's being talked about are national databases, and I think we need to engage amongst ourselves. And once we decide that we do want some national databases, I'm certain that if we go back to EBI, get their experience and get their advice, I'm sure that they will provide that. But the point is that we need to make up our minds, what kind of databases do we want them? In any case, many of us who generate the kinds of data that on international consortia, et cetera, already get deposited in EBI or NCBI and places like that. So do we need another database? But that's a separate, kind of discussion that we should hold within our country and then go back to EBI and request their help should we need one.
Yeah, I can only add to that there are different levels of databases, of course. There are databases of general importance, yeah. Everyone uses EMBL, GenBank, DDBJ for nucleotide sequences. Everyone uses PDB. Anyone uses UniProt. Yeah? So these are really top core resources, and they need to be maintained internationally in a good way. But there are databases. I don't know so much about India, so I give you an example. The Norwegians are very interested in salmon, because salmon is a big part of their economy. So they want to know everything about the genomes of salmons, and of course, their salmon database is for them nationally important, while I think you're not so much interested in salmon here, salmon genetics. So, and you will have the same. So there will be things which are globally important, regionally and on certain continents important, nationally important, and only locally important for different researchers. And for these different resources, you need to have different tracks. I think if you have a global resource and try to run that on PhD students, you are bound to fail. Yeah, that doesn't work. If that is part that you want to study a subset of protein kinases and look into their dynamics, that may be interesting for a small field of people and really great work, but it's a research project. It's not an infrastructure project. And there you need to come up where you see this scales and these divergences, and how you can form nationally the funding streams to support these different levels of activities, international, national, and more research focused.
Yeah, I think we should call off this discussion question answer session. Maybe we take a 10-minute break instead of a 15-minute break, and then come back quickly for the panel discussion. Is that good? So 11:45, we will be back.